Good morning, everyone. We are going to jump into chapter 8, which is on cell communication. Why are we exploring this chapter? Well, we want to understand how is it that a cell responds to the environment in which it is placed in. So a cell can either respond to external signals that are received by the cell, or it can be the result of cells communicating with each other. And so we're going to explore both of those possibilities, responding to external signals such as hormones, as well as responding to signals and direct interactions by other cells. In order to illustrate the impact of chemical signals and how that might modify what's going on within a cell as well as the behavior of an animal, I thought we would start by looking at these two coyotes. The one on the left is obviously in a well-fed state, and the one on the right is in a more fasted state. So the question becomes, what are the differences in chemical signaling taking place in each of these coyotes based upon their fed state? So there's an interesting signaling molecule known as leptin, which has been found to be basically a satiety factor. So for our well-fed coyote who has lots of fat stores, he is able to secrete a hormone or a signal known as leptin. And leptin is able to go from the fat into the blood supply and will signal to the brain of this coyote that he is indeed satisfied. This will indicate that his metabolic needs are being met, and so this coyote can focus on doing other things like playing or sleeping, and he can do things besides just looking for more food. The emaciated, struggling coyote, on the other hand, has a much less fat mass, and so he is going to be secreting less leptin. And thus, his signal will not be receiving that message that indeed he is satisfied. So he will be focused on doing things like foraging for more food until those leptin levels go up, which will ultimately signal his brain that he can focus on other activities. So the question is how is that leptin signal being made, and is there a gene that actually is responsible for making this chemical message? And if there is a gene that is able to make this chemical message saying that we are full and satisfied, what would happen if such a gene were mutated? What do you think would be the phenotype of mutation in the leptin gene? Again, leptin is a satiety signal, so if you don't have that satiety signal, what do you think that would look like? So go ahead and pause the video and take a second to answer that question. So hopefully you came to the understanding that if there is no leptin gene or a mutation in the leptin gene, that would lead to no satiety, which would mean you would never receive that signal to stop eating, and that actually could lead potentially to obesity. In the lab, we actually have something called a knockout mouse, in which certain genes are systematically knocked out in a mouse model system. And so there are two interesting mouse model systems that pertain to obesity. One is the DB-DB mouse, which is for diabetes, and so particular genes have been knocked out that lead to a diabetic mouse model. And then we also have another gene that, once it was knocked out, led to obesity. And so we call this the OB mouse, which means the gene has been deleted in this particular mouse. So here again we have these two mice. The one on the left is the obese mouse, and the one on the right we call our wild type mouse or a normal mouse. And the question is, what is it about the mouse on the left? What is causing it to become obese? And so the hypothesis is, if the obese mouse is obese simply because it's not receiving a signaling hormone, in this case leptin, and is never satisfied, would there be a way that we could combine the physiology of these two mice, which is a process that we call parabiosis, and actually physically connect these two mice so that the signaling molecules present in the wild type mouse are being shared with the obese mouse? And so these studies were done, and after several weeks, the obese mouse, the OB minus mouse in which that leptin gene had been knocked out, was able to obtain normal weight once it was able to receive the chemical signals that were present in the wild type mouse. So based upon these studies, what we were able to determine is that the instructions for making that chemical signal, the signal leptin, is carried on a gene, and that gene can indeed be mutated. And the chemical signal is able to travel through the blood and affect appetite, because when we fused the physiology of those two mice, the wild type and the OB minus mouse, the OB minus mouse was able to receive the signal from the wild type mouse, and normal appetite response was observed. So oftentimes students then want to know, well, is this possible in humans as well? And indeed it is. Here's looking at a study of some families. These are Pakistani families in which the leptin gene has been, is not functioning. And so you can see that this has led to obesity. There's also whole sets of families that have been observed in which the receptor for the leptin gene is also knocked out, and likewise that leads to obesity. So this entire chapter is really about understanding how it is that cells are able to communicate, and how is it that cells receive signals from the extracellular environment, and that in turn causes a series of changes within the cell so that it can respond to the signal that it just received. Now, a couple of important things to keep in mind as we are talking about cell communication. So we've spent a lot of time talking about the phospholipid bilayer and the makeup of that phospholipid bilayer. And so really the way that our cells are going to respond to the extracellular environment is going to be based upon some cell surface receptor on the plasma membrane that is going to be responding to a signal that is received at that cell surface receptor. That cell surface receptor, in turn, is going to mediate a series of intracellular changes that ultimately will cause the cell to respond in a particular way to that extracellular signal that it initially received. So even yeast cells are able to undergo this whole signaling event in response to the presence of things like glucose. So yeast cells, in order to survive, need some sort of energy input, and if all of a sudden a yeast cell finds itself in the presence of glucose, it's going to want to make rapid changes so that it can optimize on that presence of that particular nutrient. So we can look at the yeast cell, and we see that they do have something called a glucose receptor. And so the glucose receptor in this scenario is our receptor, but the ligand or the signal would be glucose itself. So what you can see is that if this cell all of a sudden finds itself in the presence of glucose, we're going to have the binding of glucose to that glucose receptor, and that's going to initiate a series of events that take place within the cell. And so if indeed the cell finds itself in the presence of glucose, what are some logical cellular responses that would take place upon binding of glucose to that receptor? Well, for one, the cell is going to want to allow for the massive intake of glucose. And so the yeast cell is going to start to express a gene that makes the protein for these transporter proteins. So these transporter proteins are going to get made, and you'll see they act kind of like ligand-gated channels, and so all of a sudden you're going to have this massive influx of glucose into the yeast cell. In addition to the glucose transporters, the cell is also going to allow for different metabolic enzymes to be expressed and activated, and they're going to be able to break down the glucose that all of a sudden has made its way into the cell. And in so doing, the yeast cell has responded swiftly to the presence of glucose that was outside the cell, initiated a signaling cascade within the cell that led to the expression of a gene that ultimately led to the expression of this particular protein, the glucose transporter, such that glucose can now be taken up from outside the cell and brought into the cell, as well as the activation and mobilization of different metabolic enzymes that can help to break down the glucose that is in the surrounding environment.